So the topic of my talk is adapting durum wheat to drought and crown rot conditions. Future projections in terms of environment, there will be increased drought in many regions of the world. Incidentally, some of those regions in brown are where we currently grow most of our durum wheat around the world. So this is really bad. Another major issue that durum wheat production faces is crown rot disease. And that is because farmers around the world have been adopting minimum tillage practices. And what we see is more stubble in the field. And crown rot disease caused by Fusarium species actually lives and survives in the previously infected stubble. So what we see in Australia in particular, a huge buildup of inoculum. And that is, again, really bad because all our durum wheats are susceptible to crown rot disease. When water is not an issue, the crop might still be showing signs of infection, as you can see down here, if it works. Oops. Yep. And, but the farmers don't experience big yield losses. And we see the crop really looking healthy, and we just experienced around approximately 5% of yield loss. It is only when high levels of inoculum coincide with drought is when we see massive yield losses, up to 89% yield loss. And what happens is actually all the stems of the plants get clogged up with mycelium, they can't suck up any more water, and they die prematurely, basically leaving us out with those conspicuous white heads. And if we look inside those white heads, we see either no grain or at best shriveled grains. As plant breeders and geneticists, we would love to introduce resistance and make those variety resistant. But unfortunately, as I said, our durums are susceptible. And also fungicides do not work. So we thought, how about a physiological approach using drought adaptive traits to minimize yield losses under crown rot conditions when drought occurs? I'm talking about drought adaptive traits that are associated with water use efficiency traits, such as transpiration efficiency, stay green, osmotic adjustment, and perhaps look underground and optimize root system architecture so that our plants have better access to the water in the soil profile and avoid drought. So as plant breeders, what do we usually do? We thought, how about creating populations to look at the value of those water use efficiency traits to minimize yield losses under such environments. So we teamed up with durum wheat breeder Dr. Filippo Bassi, based here in Icada, and set about creating populations combining the genetics of Icada lines with Australian cultivars. And the reason why we wanted to do that was because we know that Icada has been breeding for drought adaptation and tolerance to diseases for such a long period of time. And also, Australian cultivars are very well renowned for their end-use quality parameters. So we thought, why not create these populations, combining these two distinct genotyping pools down here, and perhaps look at the value of those water use efficiency traits, in particular under Australian environments. So just to give you an idea of the durum-NAM nested association mapping population that I developed, I crossed a number of Icada lines to two reference varieties, Australian cultivars, Dibia aurora and Jandaroy. And using single seed descent, created about 100 individual per population. And to speed up the whole process, because it takes such a long time, I used speed breeding, which Lee will cover that tomorrow And also, so just briefly to give you an idea what we do in the speed breeding, we expose our plants to the extended photo period, and so they go through the reproductive phase really fast. So we go from seed to seed in just 2 months. The F6 homozygote lines were genotyped using dart-seq marker platform. This is a heat map, and the more red it is, the more relatedness. So when we looked at the population structure, we found it was really like a textbook example of linked populations. So as you can see here are the two backgrounds, the reference parents, DB aurora and Janderoy. And here, the subnoun populations derived from the crosses between founder lines to each one of those reference varieties. And here are the two common founder lines that are crossed to both reference varieties. So now we have this fantastic population. So we thought, let's look at the order use efficiency traits. So remember, the order use efficiency traits are above and below ground. And here we wanted to start with root system architecture. To do that, we used ClearPot method, developed by one of my colleagues at University of Queensland, Dr. Cecile Richard. It is a really high throughput and very cheap way to phenotype large number of plants in a very short period of time, and very cheap. It's just a clear garden pot. And what we do is you can use any sort of camera, digital camera, you can use your phone and measure the root angle using ImageJ. It's a freely available software online. What we found was a large variation in the parental set ranging from 48 and 112. And that's what we really look for usually, the variation. To look at the correlation with the field, we did some of the hard works. I lost some weight. And we looked at the correlation with the field. So to do that, we set up this experiment and used shavolomics. And again, we found large variation ranging from 51 to 85 degrees. When we did the correlation between these two methods, we found really good correlation. And that lines up with what has been reported by Roberto's group. And because we were really happy with this correlation, I proceeded using the ClearPot method because it's much easier. This is why I put on some weight now. So then we wanted to track down some of the genomic regions that are controlling the root system architecture, growth, and distribution. So using the phenotypic data from using the ClearPot method for a subset of 393 nested association mapping lines and high quality markers, about 2,500 of them, I was very lucky to find this nice QTL on 6A using a Genable package. And when I looked at the correlation between the markers that are highly associated with root angle, I found that they were in high linkage disequilibrium. And what that means that is it is more likely for them to co-inherit. Using this set of markers, I constructed a haplotype network. So after doing the haplotype analysis, I found that the majority of lines were falling into two major haplotype groups. With HAP1, like with about 142 individuals carrying haplotype 1 and 120 individuals carrying haplotype 2. The difference between these two haplotype groups was 8 degrees. It was really significant, and now we might think it's not much, but we know that root system architecture angle, like the root angle, is determining and has big implication on the root system architecture further on during the crop development. This is just to give you a taste of how the phenotypes looks for each one of those haplotypes. As you can see here, those plants are, it's one of the parents actually, it's OTB4, Outro4, and the other set is the DB Aurora parent. Now then we wanted to know if root system architecture has any implication on yield under drought conditions. So we set up this experiment in one of the drought stations in Queensland, in Australia, and tested a subset of 168 individuals, collected data regarding flowering time, plant height, yield and its components, and also weekly NDVI measurements. Using the weekly NDVI measurements that I collected from flowering time up till full maturity, I was able to fit it into this logistic curve for each plot. And from this curve, which we refer to as the senescence curve, we can extract many stay-green traits, such as the area under the curve and number of days from flowering until 10, 50, and 90% of senescence, and also the rate of senescence. The key results from this really hard work, each time you walk like 12 kilometers, 15 kilometers, was that stay-green traits were significantly correlated with yield. Then we wanted to see if root angle QTL has any effect on yield performance under drought conditions. So we found that the individuals that carry haplotype 1 and comparing it to the individuals that carry HAP2, there was a significant difference in terms of the area under the curve. And what happens is that those individuals that carry haplotype 1 for the root angle QTL are senescing faster in comparison to the other one, which shifts the senescence curve and increases the area under the curve, which in another way, it's prolonging the grain filling stage, which eventually translate into massive yield losses, yield differences, sorry. So exactly what happens is the grain filling period increases and the grains fill, and this is what we see, massive differences in yield. Then we wanted to know if water use efficiency traits can reduce yield losses under crown rot conditions. So we set up another experiment in the same drought station, but this time in different environment, with inoculated with crown rot really heavily. So we had it that time for 3 years. And what we did, we collected similar data sets, but this time we scored for crown rot disease as well. So what I did, I scored for the percentage of the white heads in each plot a couple of times, and also at maturity pulled out 10 plants per plot and scored them for the stem browning. Then we came up with this severity index combining these 3 data sets. And run the genome-wide association study using the markers, the high-quality markers, and found this QTL on 6B. Incidentally, we found similar region was associated with days to flowering, plant height, and also, most importantly, stay green. Then performed the haplotype analysis for this QTL and found that there was significant differences between the individuals carried HAP1 in comparison to haplotype 2 and for the stay green traits. And that again translated into differences in yield, as you can see. Oh, I'm in the opposite direction here. Yeah. Just to summarize this slide, I found that the we found that plants are achieving this tolerance to crown rot is by modulating the canopy development and using water in a smarter way. And so, again, prolonging the grain filling period, which again translated at the end to yield. The most exciting part of this research was when we looked at the very closely related individuals that were segregating for both our QTLs, the crown rot QTL and the root angle QTL. And we compared the individuals that carried both favorite set of alleles, referred to as plus here in green, and compared them to the individuals in those sets that did not have the favorite alleles for both QTLs, referred to them as minus in orange. And what we found under crown rot conditions, there was massive difference in yield. So it was a yield benefit of 1.1 tons per hectare, and that's huge. And when we looked under drought conditions, we again, in the same station, we found a difference in yield up to 0.41 tons per hectare. And then we looked further on in conditions, in environments in South Australia and also here in Morocco, and we found similar trend. And the total so the average, the total average for these three environments was over 500,000 tonnes, and that's really great. The take-home message is, now that we have this powerful nested association mapping population, that we could study water use efficiency traits and also other traits, and that all could be harnessed using plant breeding. So we are open to any form of collaboration and sharing this material. Secondly, this measure QTL for root angle on 6A needs really further work on that. And also finding this QTL for crown rot on 6B, both these QTLs open the opportunity to combine above and below ground traits to perhaps not only minimize yield losses under drought conditions, but also under crown rot conditions. Sorry, I didn't mention the last point. So what's next? We've done this work. We would love to do even further work on these sets of drought adaptive traits in more diverse environmental context, and also would like to use more like breeding strategies such as genomic selection to compile all those favorite alleles of these water use efficiency traits for above and below ground traits. At the end, I would like to thank my supervisors and my Hiki Lab, based at the University of Queensland and Queensland Alliance of Agriculture and Food Innovation. And I would also like to give special thanks to Monsanto Bichel Borlaug International Scholarship Program, which covered most of the costs associated with my research. And also I would like to thank the University of Queensland Research Scholarship. Happy to take any questions.